Hello and welcome dear viewers to the video on impurity assessment in pharmaceuticals. In this video, we are going to consider the key points for impurity assessment so that you can have a formulation with impurity specifications which is in line to the regulatory requirements and for the product which will get less regulatory queries and the faster approval. So what are the key considerations for impurity assessment for API and the pharmaceutical formulation we are going to see. First of all, whenever you are going to study the formulation, first of all you need to understand the impurities of API. So consideration for API impurities to be done. The consideration should involve in-depth understanding about the process impurities, then degradation impurities, and some impurities which are process as well as degraded impurity. So these are the general impurities coming from process, synthesis route, intermediates, starting materials, or degradation due to aging of the API, due to some storage conditions, environmental factors, and some impurities which are process related and also the degradation type of impurities. Then coming to the genotoxic impurity, we have to understand and study whether there is any genotoxic impurity potential or any risk assessment is done for the API. Generally, this information is present in the DMF, and also as required, the API supplier or API vendor can provide this information. Then one of the most important point is nitrosamine impurities. Whether there are any nitrosamine precursors or any impurity which will be responsible for generation of the nitrosamine impurities. So that may be the process impurity or degradant impurity, which if present above the some threshold will lead to formation of the nitrosamine impurity. Then elemental impurities, residual solvents, and other impurities. So elemental impurity declaration to be provided by the API vendor, that is DMF holder or EDMF holder, nitrosamine impurity and its declaration, and if some nitrosamine impurities are present in the API, then how those impurities are going to be controlled. Then what are the residual solvents used in the manufacturing of the API and how it is controlled with their specific limits. So these are the points to be considered for API. Then the justifications and specification limits for impurities are need to be considered. How the impurities are limited and how the specifications are given for the API, that should be assessed critically. Then safety assessment for potential degradation impurities and the mutagenic impurities. If there is any risk of mutagenic impurity, then how that impurity is being controlled. API assessment should be done based on considering the formulation maximum daily dose, so that there will be a limit for API based on to the formulation daily dose. Many of the time it happens that the same API is used for formulating the different formulations like tablet, capsule, oral suspension, oral solution, and injections. So based on the formulation type, the maximum daily dose of the drug substance varies, and based on that variation in the maximum daily dose, the limit of the impurities should be given. For example, if the maximum daily dose of IR formulation is 20 mg per day, then the same API may be formulated with 30 mg as a single oral formulation with extended release design, or with the IV route of administration with around 1 gram maximum loading dose per day, depending on the patient condition or the patient requirement or the maximum loading dose per day. So the same API may get formulated into different formulations and can be administered with different routes. So that point should be considered when you are going to study the API related impurities. The last example of tablet, IR tablet, extended release tablet and injectable formulation I have given just for example. But whenever you are going to consider the API impurities, that time this type of questions should be assessed and their answer should be find out, so that you will understand whether the API being used by the formulator or formulation, that API is being used by the other applicants or for making the other formulations with different route of administrations. Then safety assessment for potential degradation impurities and mutagenic impurities is to be provided. Then also consider whether there is a polymorphic form conversion, whether there is polymorphic form retention in the API, and if the form get changed, then how it is going to impact onto the impurity formation and the impurity specification. For example, if the API exists in number of polymorphs and also it is available in amorphous form, that time if the amorphous form get converted to crystalline form or crystalline form may get converted into amorphous form, or crystalline form may get changed to other crystalline form, that time how it is going to impact the impurity profile of the API and how it will relate to the formulation. That points are required to be considered. When you are going to consider the generic product, that time always have the in-depth understanding about the reference product MDD, that is maximum daily dose. The maximum daily dose is the dose which is administered to the patient in the time period of 24 hours. So consider the indications as well. Consider the dosing regimen based on to the patient condition and requirement. Many of the time this happens in the formulations that 2 tablets are given per day, as 1 tablet after meals and 1 tablet after dinner. But if in the hospital setting, in the critical condition and requirement by the patient, the innovator or reference product patient information reflect suggest to use more tablets. For example, 6 tablets or 10 tablets per day. So that time that per day dose which is maximum becomes the maximum daily dose. Then indications. What are the indications? What are the strengths? And what are the indications for each strength of the reference formulation? Sometimes the indications and maximum daily dose varies with the indications. Then what is the patient population for the reference product? Whether it is pediatric, whether it is adults, whether it is geriatric population, and duration of the treatment. So based on the duration of treatment also the impurity limits varies. Acute treatment or chronic treatment. So based on the acute treatment and chronic treatment, that can be divided into the number of days for the treatment or dose exposure, and based on that the impurity limits can be designed. So always have an understanding about the duration of treatment, and duration of treatment is very much important for justification and for specification of the genotoxic impurities. In genotoxic impurities, as per ICH M7 guideline, the exposure time is considered for giving the specifications. If you are giving the formulation for 1 week or 1 month or 1 year or 10 years, based on that the specifications are derived for genotoxic impurities. The TTC based approach is there with TTC limit of 1.5 microgram per day, and this TTC limit can be changed based on the duration of treatment. Also, there is a consideration for less than lifetime approach, LTL approach, for genotoxic impurities. So, duration of treatment is very much important. Also, route of administration is very critical to understand, whether the formulation is given by oral route, whether it is also given by the IV route, and whether the same drug substance can be formulated or is being formulated into different formulations for different routes and the disease conditions. So other formulations with the same API should also be considered. Then coming to the API related considerations, once again API form of the formulation, which API form is being used into the formulation. Generally crystalline forms are stable compared to the amorphous form. So form plays the critical role, and form is the polymorphic form or crystalline form. Then what is the intrinsic stability of the form? What are the degradation pathways and degradants? Then storage sensitivity, whether there is a sensitivity of the drug substance for the environmental factors like temperature, humidity or light. Then what is the composition of the generic product? Whether there is any stabilizer or any acidic basic material which may have impact onto the API stability and in turn on the formulation stability. Whether there is any compatibility or incompatibility of the API with the excipient or a raw material. Whether there is any interaction between the two APIs if you are working onto the combination type of formulation, that to be considered. Then what is the manufacturing process? Whether it involves use of heat, use of solvents or use of mechanical force, that should be taken into consideration. Then impurity assessment should be done based on the above points. Listing should be done for known, unknown and total impurities, and these all the impurities should have justifications and should be given based on to the regulatory requirements, ICS threshold limits for reporting thresholds, identification threshold and the qualification thresholds. Also the real-time stability data should be considered, and number of package for the same formulation should be considered so that you can select the best package in which the impurity are less. Impurity specifications are made based on the consideration for regulatory guidelines like ICH guidelines are there, European guidelines are there, ANVISA guidelines are there, Swiss Medic guidelines are there, and Japan guidelines are there, US FDA guidelines are there. So many guidelines are there which have the regulatory requirements for specific impurities and for the justification of the impurity limits. Basically, the ICH guidelines are considered whenever the impurity justification and impurity specifications are given. So the impurities are also dependent onto the critical material attributes of API critical material attributes of excipients like a level of formaldehyde in some excipient like starch, level of peroxide in some excipients like povidones. So these CMAs should also be considered because these impurities coming from the excipients can also trigger the impurity formation. Then critical process parameters like temperature plays the critical role, drying time plays the critical role, and hold time plays the critical role in impurities. That's why CMAs and CPPs are to be well understood and well defined for their impact onto the impurities. Then other factors like packaging material factors, storage related factors are to be considered. Now, if you have set the impurity limits based on the ICH requirements and regulatory requirements, but if you found that the impurities are being generated and these are crossing the limits of qualification, so then the qualification process comes into picture, and qualification is done based on the literature search. You have to provide the specific literature which gives the clear understanding about the impurities and their safety. Literature based carcinogenicity and bacterial mutagenicity potential can be determined. So some of the time the carcinogenicity and mutagenicity data is to be given for qualification. That time these mutagenicity studies are performed to qualify the impurity. Metabolite data can be used as a base for uh, qualification of specific impurities if the impurities are degradant as well as the significant metabolite. If you come to the understanding that the impurity A is a significant metabolite and it is the degradant also. So based on the metabolite data and the reference listed product or RLD or reference formulation literature, you can qualify that impurity. Then QSAR relationships can be studied, the models can be selected which are fitting for that molecule. The model fit is required to be determined. So based on the QSAR and structure activity relationship base, the impurities can be qualified. Then bacterial mutagenicity studies are performed, like AIMS test is there, which is bacterial reverse mutation assay. These are performed to understand whether there is any mutagenic or carcinogenic potential in the identified impurity. So whenever qualification activity is performed, first the impurity is required to be identified, isolated, its structure is required to be made known, and then the qualification activity is performed. Then coming to the general toxicity studies. So if the uh, impurity is not genotoxic or if it is not carcinogenic, then you can go for the general toxicity studies. And general toxicity studies are performed based on to the acute and repeated dose toxicity studies. So these studies comes under the general toxicity studies. If you found that the impurity is above the qualification level and it is not mutagenic, then need to be studied for the general toxicity studies. So rodents are used for these studies for performing the general toxicity studies, and sometimes the animals are used. So based on these assessments, key considerations, the formulation and API specification can be given, and the key points are required to be considered so that the regulatory queries will be less. Sometimes because of the inadequate justification for impurity limits and impurity data, insufficiency of the impurity data, the generic application can get rejected, or for the generic formulation it becomes very difficult to justify the impurities. So whenever you are working onto the generic formulation, that time from the start of the formulation, the impurity assessment should be started, and these impurities are required to be critically monitored. The factors which are leading for the impurity formation and increase in the impurity levels should be understood in a scientific way. So this is for the impurity assessment, and I hope uh, I have covered the key points. The impurity assessment is also a big topic and cannot be covered in one video. So watch the videos which I have earlier posted so that you can have the good understanding about the topic. Thank you for watching the video. Please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.